All right, today we are diving deep into a field that is completely changing how we understand biology and medicine. It's called proteomics, and really, it's all about learning to speak the language of life itself.
But first, I want you to just imagine something. What if, right this second, we could press pause and take a snapshot of everything happening inside your body? And I mean everything. Seeing every single tiny protein, all the little machines doing all the work, and knowing exactly what each one is up to. I know, it sounds like pure science fiction, doesn't it? Well, that science fiction is quickly becoming science fact. And the key to making it all happen is this incredible field called proteomics. It's our first big step in this whole decoding process. So what is it, exactly? To put it simply, proteomics is the large-scale study of proteins. We're not talking about looking at just one protein at a time anymore. No way. We're looking at the entire collection of proteins, what scientists call the proteome, all at once, in a single go. You know, it's kind of the difference between watching one car drive down a street versus seeing the traffic flow of an entire city during rush hour. And when we're looking at this bustling city of proteins, we're really trying to do two main things. First, identification. We need to know who's actually there. It's like taking attendance. And second, quantification. How many of each protein are there? Do we have a ton of one and just a few of another? Getting the answers to just those two questions, well, that's absolutely crucial. Now, I know what you're thinking. How on earth do you see, let alone count, things that are so small they're basically invisible? Well, to do that, scientists had to invent a pretty special and very powerful kind of engine. And that engine is called Mass Spectrometry, or just MS for short. And let me tell you, this thing is the absolute workhorse of the entire field. Almost every single discovery we're about to talk about, it's only possible because of this incredible machine. You know, the easiest way to think about a mass spectrometer is like a ridiculously precise scale, but for molecules. What it does is measure something called the mass-to-charge ratio. So imagine you take little fragments of a protein. You give them a little electrical zap, a charge, and then you see how they fly through a magnetic field. Based on their flight path, the machine can calculate their exact mass. And because these fragments have unique masses, it's like giving each one its own molecular fingerprint. The whole process is really like a three-step dance. First, you have to give those protein fragments that electric charge. That's ionization. Then, the machine uses magnets to sort all of those charged bits and pieces by their mass. That's the mass analysis. And finally, a detector at the end counts how many fragments of each specific mass hit the target. That's detection. Simple as that. But here's the thing. Getting all those numbers out of the machine, that's just step one. What you get is this massive, incredibly complex stream of raw data. The real challenge comes next, turning all that noise into something that actually means something. And this is that whole journey. The raw data has to get cleaned up, filtered, that's all pre-processing. Then you've got this super powerful software that has to identify all the little fragments, or peptides, by matching their masses to known sequences. And finally, the computer pieces all of that information back together, like a giant jigsaw puzzle, to figure out which proteins were there and in what amounts. It's a huge computational lift. So once scientists solved this puzzle and they've identified a protein, where does all that super important information go? I mean, it can't just sit on one person's computer, right? It has to be shared with the rest of the world. And that is exactly what the Protein Data Bank, or PDB, is for. Think of it like a massive, global, open-access library. It's packed with tens of thousands of protein structures, and anyone, anywhere, can access it for free. It is truly one of the cornerstones of modern biology. So, inside this digital library, researchers can pull up the 3D atomic blueprint of a protein. They can visualize how it folds and moves. They can even use computer models to predict how it might react to a brand new drug. It's just an absolutely essential tool for making new discoveries. And these structures aren't just guesses either. They're the result of some seriously powerful techniques. Things like X-ray crystallography, NMR spectroscopy, and the more recent Nobel Prize-winning cryo-electron microscopy. These are the methods that let us see these molecules in such unbelievable atom-by-atom detail. Okay, so we've got the tech and we've got the global library. But how does this all connect back to something that matters to every single one of us, our health? Well, this is where proteomics really starts to shine. This is the key. By comparing the proteomes of healthy people to those with a disease, scientists can pinpoint the exact proteins that are causing the trouble. This helps us find new drug targets. Take breast cancer, for example.
Scientists found that some cancers have way too much of a protein called HER2. That discovery led directly to a life-saving drug, Herceptin, that specifically goes after that one protein. It also lets us find biomarkers in the blood, early warning signs, to catch diseases like cancer way sooner. And all of this leads to the ultimate goal: personalized medicine, where treatments are designed for your specific biological makeup.
This is already changing so many lives, but the field is just moving at a breathtaking pace. The technology gets faster, it gets cheaper, it gets more powerful every single year. So what's coming up next on the horizon?
Well, the future is all about getting more integrated and more precise. We're moving towards what's called multi-omics, where we combine proteomics with genomics to get a full 360-degree view of biology. We're also developing single-cell proteomics to zoom in from just a tissue sample to see the proteins inside one single cell. And of course, we're using AI to find patterns in these mountains of data that a human could never, ever spot on their own. Of course, these huge leaps forward bring huge challenges. I mean, how do you even store and analyze these mind-bogglingly massive data sets? How do we make sure every result is perfectly accurate? Solving these problems is exactly what's pushing the next wave of innovation. And that really brings us right back to where we started. We began with that what-if question, and we've seen how proteomics is turning that into a reality. So as this window into our own biology gets clearer and wider, you just have to wonder, what other profound secrets about disease, about aging, about life itself are our proteins just waiting to tell us?